Um, okay, so let's move on to our next speaker, who is Dr. Richard Francis, and I will read a little introduction here. Sorry, my materials in front of me. So Dr. Francis is an MD PhD associate professor in the Department of Pathology and Cell Biology at Columbia University in New York. He's the director of the Special Hematology and Coagulation Labs at New York Presbyterian Hospital, Columbia University Irvine Medical Center, where he's also the assistant program director for the residency training in clinical pathology. He's an adjunct associate professor at Rutgers in Newark, New Jersey. Dr. Francis is board certified in clinical pathology, blood banking, and transfusion medicine, and has published on a variety of topics, including hematology, hemostasis, blood collection, and transfusion. His research has mainly focused on the contribution of oxidative stress to the pathophysiological changes in red blood cells that occur during refrigerated storage. And he's published some very interesting work recently, and I asked him to come talk about it. So, Richard, over to you.
Great, thank you so much. I appreciate that introduction, and I appreciate the invitation to come and share some data. Can you see my screen in slideshow? Okay, perfect.
Yeah, great.
So, so this audience doesn't need this, but I will still let you all know, of course, that red blood cell transfusion is a widely used therapeutic intervention, and it's the most common intervention for hospital inpatients here in the United States. And it's the mainstay of therapy for sickle cell disease and also for thalassemia major patients. Of course, the majority of red blood cells that are transfused are collected, stored in the refrigerator for some period of time. And we understand that there's this collection of changes that occur to the red cells, both in morphology, as you can see here, and comparing what red cells look like at day 0 versus at day 42. And also many biochemical changes that occur in the red cells as they sit in the bag in the refrigerator. And together, these lead to decreased red cell survival in vivo. So the FDA criteria for a stored red cell product, in order for a red cell product to be stored for up to 42 days prior to transfusion, the mean 24-hour red blood cell recovery should be greater than or equal to 75%. The standard deviation for this study should be less than or equal to 9%. Out of 20 studies that are performed, no more than 2 should be less than 75%. And there should be less than 1% of hemolysis, spontaneous hemolysis in vitro. And so for doing these 24-hour post-transfusion recoveries, what we do is we collect a unit of blood from someone, we store it in the refrigerator for, let's say, up to 42 days. At the end of that time, we take an aliquot of around 20 to 25 mils of blood, centrifuge, wash, incubate with radioactive chromium-51, and then wash again. And then we give an IV push of this labeled blood product and then take several blood draws over time over the next 24 hours in order to calculate the percentage of red cells that are surviving after 24 hours. And so here in the red is an example of a really good post-transfusion recovery of greater than 90%. And then here in blue is one that's roughly around 75%. And so this has been done, and here's a pretty famous paper from Dumont, where this was done in many, many donors, and it was found that, yes, you have quite a nice distribution of the red cell survivals, the 24-hour post-transfusion recoveries. Here's 75%, your cutoff for the FDA criteria. And you find, of course, that there are some people who have recoveries that are less than 75%, and we consider them maybe poor storers. And then there are those who have red cell recoveries that are way above 75% and who are often referred to as being good storers. One of the things that we know happens during storage is that we have the accumulation of reactive oxygen species and oxidative damage over, let's say here, the 6-week storage period. And so that raises the question of how do red cells handle oxidative stress in any scenario, but in this case, let's say in the scenario of blood storage. And so there are several pathways that red cells have, or that cells have in general, for dealing with oxidative stress. So one is the peroxyredoxin pathway that helps to detoxify hydrogen peroxide. We have also catalase that also acts on hydrogen peroxide. And we also have the ascorbate and glutaredoxin pathway. One common pathway or one common element that is involved is glutathione. And interestingly, if we talk about glutathione, or in this case, especially reduced glutathione, there have been studies to show that reduced glutathione stability correlates with 24-hour red blood cell survival or 24-hour post-transfusion recovery. So we might think about this a slightly different way and say that, well, maybe blood donors who have poor 24-hour recoveries, maybe their red cells are oxidative stress susceptible, and possibly blood donors who have very good red cell recoveries, well, maybe their red cells are oxidative stress resistant. So glutathione or reduced glutathione is used as a substrate by glutathione peroxidase in order to convert hydrogen peroxide to water. In the midst of this reaction, we end up oxidizing the glutathione, and so this glutathione then needs to be recycled back to the oxidized glutathione needs to be recycled back to reduced glutathione, and NADPH is used as a substrate by glutathione reductase in order to reduce oxidized glutathione back to reduced glutathione. Glucose 6-phosphate dehydrogenase is important for regenerating the NADPH that is needed for the glutathione reductase reaction. So G6PD uses glucose 6-phosphate and NADP plus in order to make 6-phosphoglucanolactone and NADPH. So there is a disorder, glucose 6-phosphate dehydrogenase deficiency, which is a genetic disorder in which red blood cells respond poorly to oxidative stress. And so just to remind you, G6PD is important for producing the NADPH that we need in order to help to reduce glutathione. So G6PD deficiency affects approximately 400 million people worldwide. It is the most common enzyme deficiency among human beings. The risk of hemolysis in response to, it causes a risk of hemolysis in response to oxidative stress. In the United States, the population that is typically most affected are males of African descent who have a frequency of approximately 10%. Importantly, there's no routine screening of blood donors, and refrigerated storage is considered to be an oxidative stress. So this leads us to the question, is it possible that glucose 6-phosphate dehydrogenase deficiency among blood donors contributes to the variation in storage quality that we see? And why is this important? So we did a study where we looked at the frequency of G6PD deficient red cells in the inventory of our hospital transfusion service, and found that in the general inventory, the frequency is quite low at less than 1%, so 0.3%. However, when we looked at the red cells that were blood group phenotype matched for patients who had sickle cell disease, which by doing that, you're selecting for donors of African descent, the frequency was significantly higher at 12.3%. And so why might this be a problem? Of course, increased clearance of G6PD deficient red cells by patients who have sickle cell disease can lead to increased transfusion requirements, could increase the risk of iron overload, and can also increase complications that are due to hemolysis. And indeed, this has been seen where we had a study where we looked at pediatric patients with sickle cell disease who were receiving simple transfusions, and we looked at the hemoglobin A between the change in the hemoglobin A between two transfusion episodes, and looked at a bunch of different factors to see, which correlate with this change in hemoglobin A, this decrease in hemoglobin A between transfusion episodes. And as you can see here, there was a significant correlation with getting a G6PD deficient unit and the change in the hemoglobin A in between transfusion episodes. And as you can see, several factors were examined here, and the most significant correlation was with having received a G6PD deficient red cell unit. The patients who received the G6PD deficient red cell unit also had higher reticulocyte counts in between their transfusion episodes. Again, suggesting that there's a decrease in the efficacy of those transfusions. So we set out to evaluate the effect of G6PD deficiency on red cell storage quality in a more systematic study. And so we screened 145 males for G6PD deficiency, and we were able to recruit 10 G6PD deficient donors and 30 G6PD normal donors. These participants all donated 1 unit of leukocyte-reduced red cells, and these red cells were stored in AS3 additive solution for 6 weeks. And so during this 6-week period, every week we removed a sample from the unit and tested for and saved the sample for metabolomics, and also did some more conventional biochemical measures during this time. And at the end of the 6 weeks, we performed a 24-hour chromium labeled red cell post-transfusion recovery study. So of the 145 subjects that we screened, we had 130 who were G6PD normal, 15 who were G6PD deficient. We had 30, again, normals who donated blood, 10 deficient who donated blood. We had 3 people who were lost from this study from the normal group. And so we ended up with 27 individuals who completed the study for the G6PD normal arm and 10 from the G6PD deficient arm. And so this just shows you the range of G6PD activities among our participants. And so here you can see we have a clear separation when it comes onto the G6PD activity between those who are normal and those who are deficient. And of the G6PD deficient blood donors, 1 had the Mediterranean variant, which is a class 2 more severe variant. 9 had the A-, which is the most common G6PD variant and is expected to give a more mild deficiency. It's a class 3 variant. And then here you can see our G6PD normals. So the first thing we saw when we looked just at baseline values, so before storage in deficient and normal donors, was that G6PD deficient subjects seem to have a compensated chronic hemolysis at baseline. And so what you can see here is that looking at G6PD normal versus G6PD deficient, there was no difference in the hemoglobin, the average hemoglobin, and the hematocrit between these 2 groups. However, the red cell count was significantly lower in G6PD deficient individuals, and the reticulocyte count was significantly increased in G6PD deficient donors, suggesting this sort of subclinical hemolysis, if you will. Then we looked at some of the metabolomics for these donors and found that overall in G6PD deficient red cells during at baseline, that there seemed to be an increase in the glycolytic intermediates in these red cells. And so if you look here, you'll see that for certain analytes like 2,3-phosphoglycerate, ATP, and also lactate, the G6PD deficient donors or G6PD deficient red cells at baseline were significantly higher. And this is not unexpected because, as you know, there's basically this bifurcation here. So glucose becomes phosphorylated, so we have glucose 6-phosphate, and then glucose 6-phosphate now can either enter into the Pentos phosphate pathway via G6PD, or it can continue on through glycolysis. So it would be expected that if there's a problem with the Pentos phosphate pathway, that this glucose 6-phosphate could very easily just be used by glycolysis. So there's a very intimate relationship between glycolysis and the Pentos phosphate pathways. And so again, starting out here, we have glucose, which is made into glucose 6-phosphate, and then glucose 6-phosphate is converted by G6PD to 6-phosphoglucanolactone, and then 6-phosphoglucanolactone is converted to 6-phosphoglucanate, and this is considered the oxidative phase of the Pentos phosphate pathway. Under the activity of 6-phosphoglucanate dehydrogenase, we then derive ribulose 5-phosphate, and then ribulose 5-phosphate is then converted to ribose 5-phosphate. And once we get to ribose 5-phosphate, we're on into what's called the non-oxidative phase of the Pentos phosphate pathway. And the intermediates that are here, particularly ribose 5-phosphate, are very important for then entering into the purine salvage pathway, which is important for maintaining, for for maintaining the concentration of ATP within cells, within red cells. Important here is to realize that the non-oxidative phase of the pentose phosphate pathway is connected to glycolysis by several reactions that allow intermediates to go back and forth. So therefore, the point here is that intermediates of the non-oxidative phase can either come from the oxidative phase, which is for which G6PD is important, or they can be contributed from glycolysis. So we did tracing experiments to examine the activity of the pentose phosphate pathway in both G6PD normal and G6PD deficient red cells. And what we did was you're able to treat red cells after labeling with isotope labeled glucose, treat the red cells with methylene blue in order to activate the pentose phosphate pathway, and then you can look at the range of products that you get, which will tell you whether whether the pentose phosphate pathway was used or glycolysis in order to derive those products. And if we look here, what we see is that G6PD normal red cells, when given methylene blue, actually did show an increase in the pentose phosphate pathway. However, G6PD deficient red cells did not, as expected. Next, what we were able to do is we were able to look at the pentose phosphates that we see in the non-oxidative phase of the pentose phosphate pathway and determine whether they were derived from glycolysis or were they derived from the oxidative phase of the pentose phosphate pathway. And what we can see here is that in G6PD deficient red cells, these pentose phosphates were largely derived from glycolysis and not from the pentose phosphate pathway, as we would expect, simply because of the lack of G6PD activity. So what we found so far at baseline is that G6PD deficient subjects demonstrate a compensated hemolysis. There seems to be an increase in glycolysis that is accompanied by a defect in the oxidative phase of the pentose phosphate pathway. And the pentose phosphates that are needed for purine salvage are largely provided by glycolysis in G6PD deficient red cells. So now we look to see what was happening during storage. And so here, if you look at this principal component analysis plot, what you can see here is that there seem to be changes in metabolites during storage that were related to storage time and were also related to G6PD status. So again, as we saw at baseline, when we look here at some of the glycolysis intermediates during storage, you can see that G6PD deficient red cells had increase again in glycolytic intermediates. And so in these graphs, when you see the symbols that are red with the open circle, that's actually our one Mediterranean variant or more severe G6PD variant, so that you can see in the different slides. So here, increased levels in 2,3-phosphoglycerate and increased levels of pyruvate. Interestingly, during storage, ATP levels were maintained in G6PD deficient red cells, basically the same as they were in G6PD normal. So there is no increase or accelerated decrease of ATP levels in G6PD deficient red cells. And this is important because we know that ATP and total adenylates have been shown to correlate with PTR, and that higher ATP levels correlate with better red cell viability. So how is it that the G6PD deficient red cells are able to maintain their ATP during storage? And again, we go back to the idea of this non-oxidative phase of the pentose phosphate pathway, where of course important in this is ribose 5-phosphate, which is converted to phosphoribosal pyrophosphate, which then enters into various reactions for purine salvage that, if everything goes well, will lead to the production of ATP, and if it is ineffective, will lead to the accumulation of hypoxanthine. So G6PD deficient red cells during storage are actually able to maintain they're pentose phosphate intermediates. And so if we look at a few of these intermediates like ribose 5-phosphate, acetoheptulose phosphate, and erythrose 4-phosphate, you can see that the G6PD deficient red cells continue to maintain throughout storage these intermediates. And again, as we saw before, the ability to maintain this is through glycolysis. So if these are the intermediates, and this is basically the entrance of these intermediates from glycolysis, and up top here would be the oxidative phase of the pentose phosphate pathway, the majority of these are going to be contributed from glycolysis. So that increased glycolysis helps the red cell. So deficient G6PD deficient red cells show increased glycolysis throughout storage. The decline in ATP throughout storage is similar in deficient and normal red cells. And again, the levels of the pentose phosphates needed for purine salvage are maintained throughout storage in G6PD deficient red cells. So we go back now to looking at glutathione. And so again, I showed this earlier, glutathione or reduced glutathione stability correlates with red cell survival. And what we found here overall in G6PD deficient red cells, as one would imagine, is that there were several defects in NADPH-dependent pathways. And so one of the most important ones here for what we're talking about is the reduction of oxidized glutathione to reduce glutathione, which requires NADPH. And so if we look over the storage period, you can see that there was consistently a significantly decreased amount of reduced glutathione in the G6PD deficient red cells compared to the G6PD normal red cells. This was also seen in other NADPH-dependent reactions. So for example, the production of hexosugar alcohols from glucose is an NADPH-driven process. And as you can see here, the concentration of hexosugar alcohols was significantly decreased in G6PD deficient compared to G6PD normal red cells throughout storage. And also, if we look here at the reaction of the production of dihydroxynonene from hydroxynonenal, you can see again, which is NADPH dependent, that we have consistently decreased levels of DHN in the G6PD deficient red cells. So in conjunction with what looks like a decrease in NADPH-driven reactions in G6PD deficient red cells, we see an increase possibly in reactions that will produce NADH or that will also produce NADPH, right? Which both act as reducing equivalents. So here, the production of alpha ketoglutarate from isocitrate is an NADH producing reaction. And we can see here that throughout storage, G6PD deficient red cells have an increased amount of alpha ketoglutarate. And also here, the production of pyruvate from malate is an NADPH producing reaction. And as you can see here throughout storage, we have increased levels of pyruvate in the G6PD deficient red cells. Interestingly, with this increase in pyruvate throughout storage, there's not an accompanying increase in the concentration of lactate. And in general, lactate is produced from pyruvate by the activity of lactate dehydrogenase, which is a reaction that usually would consume NADH. And so what we end up here is somewhat of some people will call a signature of G6PD deficiency, is that there is this increased pyruvate to lactate ratio. So we're making the pyruvate, but we're not converting the pyruvate to lactate as one would expect. And so this led to the idea that there's possibly a diversion of NADH from being used by lactate dehydrogenase to methemoglobin reductase. And of course, this could be useful for the red cell, because if the red cell has increased oxidative stress, you will get more oxidation of hemoglobin. And so having greater methemoglobin reductase activity could be helpful in order to reduce the oxidized hemoglobin. And so somewhat consistent with that, when we looked at methemoglobin levels in the G6PD deficient red cells over storage, we did see that there was a trend towards lower hemoglobin levels in the G6PD deficient red cells over storage. and that at some points this decrease was significant. So the lack of NADPH production caused by G6PD deficiency affects various NADPH-dependent pathways, as one would expect. Glutathione is significantly decreased in G6PD deficient red cells throughout storage, and G6PD deficient red cells may have increased activity in reactions that produce NADH and NADPH as a means of compensating for the lack of G6PD activity. So this group had looked at ROS levels in G6PD deficient and normal red cells throughout storage, and found that, interestingly, the level of reactive oxygen species was basically the same. There was no difference. However, when you looked at the amount of oxidative damage in the red cells, you found that there was a greater amount of oxidative damage within the G6PD deficient red cells. And consistent with this, the same group also looked at proteasome activity in stored red cells, and looked at G6PD deficient and G6PD normal red cells, and found that there was an increase in the accumulation of proteasome activity at the red cell membrane throughout storage in G6PD deficient red cells compared to G6PD normal red cells, which might go along with the idea that there's an increase in protein damage and increase in oxidative damage, and so the proteasomes need to deal with this. So consistent with this, when we did our post-transfusion recovery studies, we did find that G6PD deficient red cells did have a significantly decreased 24-hour post-transfusion recovery. And what we also found was that whereas there were no PTR failures, so this is 75%, again, the FDA criteria, there were no failures in the G6PD normal group. However, we had 3 out of 10 failures in the G6PD deficient group. And so if you were looking at G6PD deficient red cells, let's say as a product to be approved by the FDA, by these results alone, it would fail and they would not be accepted as a product. So while G6PD activity or G6PD status did correlate with post-transfusion recovery, if you took G6PD deficient donors and took G6PD normal donors and tried to predict the post-transfusion recovery by the G6PD activity, you would not be able to. So when you adjust for G6PD status, the activity does not predict the PTR. Finally, in looking at in vitro or spontaneous hemolysis, there was no difference in vitro hemolysis between G6PD deficient and G6PD normal red cells. And so through work of the REDS3 group, similar results were actually found, or at least this was confirmed in their genome-wide association studies, where they found a correlation between G6PD status, or the presence of a certain G6PD SNP, and oxidative hemolysis. So again, in a large donor population, showing that G6PD may have something to do with the red cell storage quality. We then finally looked at metabolites and correlated them with post-transfusion recovery and tried to figure out, so what metabolites correlate with post-transfusion recovery? And what we found were that metabolites that were involved in NADH-producing reactions, purine salvage, glutathione metabolism, NADPH-dependent reactions, and also metabolites that are involved in lipid remodeling had the best correlation with post-transfusion recovery. So knowing this, a few questions come up, and 2 of the questions that I highlighted here are, does the choice of additive solution affect the storage characteristics of G6PD deficient red cells? And secondly, how did the PTRs of G6PD deficient red cells at 21 and 42 days of storage time compare? So back in 1970, the original studies that were done looking at post-transfusion recovery of G6PD deficient red cells were done by Relina. And in this study, interestingly, they stored the G6PD deficient and the G6PD normal red cells in 2 different anticoagulants. So they used ACD adenine and stored for 35 days, and they also used CPD adenine and stored for 35 days. And what they found was that there was a significant difference in the post-transfusion recovery for G6PD deficient red cells, depending on which storage medium was used. Interestingly, this difference was not found in G6PD normal red cells. So whether you stored an ACD adenine or CPD adenine, at least in this paper, did not matter. Other work from the REDS3 group actually showed that yes, there is a difference in storage based upon the additive solution. And here what they show is that there's an increase in oxidative hemolysis when red cells are stored in AS1, additive solution 1 versus additive solution 3, and this holds throughout storage. And also that there's a more rapid decrease in reduced glutathione in red cells that are stored in AS1 compared to AS3. So the additive solution may make a difference. And of course, for red cells that already have a problem with dealing with oxidative stress, this could possibly exacerbate the storage lesion. So we're planning to do a study in which we evaluate the effects of storage duration and additive solution on G6PD deficient red cells, in which we will recruit G6PD deficient subjects. They will donate blood, and the blood will be stored in additive solution 1 in this case. And then we will store for 21 days and perform a 24-hour post-transfusion recovery study and also metabolomics. And also we'll continue to store until 42 days, and then we'll do another post-transfusion recovery study and metabolomics and see what those results show. So in conclusion, G6PD deficiency adversely affects red cell storage quality. Metabolites related to purine salvage, glutathione metabolism, NADPH-dependent reactions, NADH-generating reactions, and lipid remodeling correlate with PTR. Metabolomics confirms defects in NADPH-dependent pathways in G6PD deficient red cells. G6PD deficient red cells show metabolic alterations that support the production of non-oxidative phase Pentos phosphate pathway intermediates to support purine salvage. And the choice of storage solution may further affect the storage quality of G6PD deficient red cells. And I would just like to quickly acknowledge everyone who was involved in this study, the study participants, members of the lab where I am, the Laboratory of Transfusion Biology at Columbia, our colleagues in nuclear medicine, the New York Blood Center, who helped us collect all of these units, our blood bank staff, and Angelo D'Alessandro, who did all the metabolomics, Jim Zimring, who was definitely great to talk to and exchange ideas with, and the people who helped us with our funding, the Harold Amos Medical Faculty Development Program of the Robert Wood Johnson Foundation, and also the National Blood Foundation. So thank you very much.
Thank you, Richard. That was great. So I'll start out with the first question. So the sort of first place this takes me is, should we as blood organizations be screening all of our donors for G6PD deficiency? It sounds like we probably should.
Right. So that's always the sort of the question that comes up, and it always the question is that necessary? And so when we look back at the data for what we find in the general inventory, and of course it's going to depend on your donor population. So so in our inventory, it's very infrequent. And at the end of the day, in addition, the majority of people who get, a single unit transfusion that happens to survive a little bit, less than optimally, is that really going to be a big deal? But, when you have a patient population that is going to be at risk for getting red cells that are G6PD deficient, that's where it would become useful. And the World Health Organization actually in their bulletin that gives information or guidance on selecting blood donors, they actually do say if you have a population where there is an increase in the frequency of G6PD deficiency, they do suggest having some kind of plan in place for evaluating the donors. So whether it be adding a question to the questionnaire or actually adding G6PD testing to what you do, it would seem to be prudent in certain situations.
Yeah, it feels to me like there should be at least some subset of your blood donors that have been tested. So when you're preparing products that are going to the chronically transfused, then you're not you're not contributing to additional iron overload, for example.
Right, I agree.
Yeah. Okay, Wayne, I see you've got a hand up. Go for it.
Thank you for this very comprehensive presentation, Dr. Francis. I have a question regarding the oxidative stress. So could you speak to a little bit about what are the major sources of oxidative stress in cold storage?
Right. So I think one of the major sources of oxidative stress that occurs during storage is just the idea that hemoglobin itself becomes, you know, the oxygen saturation of hemoglobin goes up. So very soon, so if blood is collected at roughly, you know, 60% oxygen saturation, within a few days to, you know, a couple of weeks of storage, you're already up to, you know, 90-something% oxygen saturation. And so the idea is that just having all of this oxygen around, and as oxygen is cycling off and on hemoglobin, within those reactions, you simply end up getting oxidative damage. And of course, with the red cells being stored at 4 degrees, you know, you have some decreased activity of some of those enzymes like the methemoglobin reductase, which allows the oxidative stress to occur. And you know, there is some evidence that if you were to take red cells and store them in under hypoxic conditions, that this can decrease the amount of oxidative damage and actually can improve the post-transfusion recovery.
Thank you.
Thanks. Again, you can put questions in the chat box if you don't feel brave enough to put your hand up.
So I see a question from Jill, and she's asking very correctly, of course, you know, did we account for the coexistence of sickle trait? And so yes, so part of what we did was when we selected our donors, when they first came in, we tested them for G6PD deficiency, and we then also did of course a CBC to make sure we excluded things like thalassemia, and we did hemoglobin HPLC to exclude again things like beta thal and sickle trait. So all of these donors were screened and none of them had sickle trait.
Thanks. Question, is the G6PD status of the recipient also important in the post-transfusion recovery comparisons?
Right, and so that's one of the limitations of the studies, that we don't know that, and that's a great point. And so yeah, so we really don't know. It could be that there's something also that has to do with, you know, the way that G6PD deficient individuals who receive red cells will clear those red cells. You know, could it be, you know, differences in the macrophage activity or something like that? And so so that is definitely a variable that that could have an impact, or that we don't know what its impact could be.
Okay, do we have other questions? If not, thank you very much, Richard. That was really great.
Great, thank you so much.